Hello. Please hit like button and subscribe my channel. Also press bell icon for future video notifications. Thanks.
SYNGAP1 helps neurons eliminate old synapses and form new ones after a novel experience, left and center left. A process weakened in mice missing a copy of the gene, center right and right. Courtesy of Gavin Rumbaugh Even, partial loss of the autism-linked gene SYNGAP1 impairs the brain's ability to respond to sensory experiences, according to a new study in mice. To encode new information about a sensory experience, a neuronal ensemble needs to redistribute its activity. Some neurons ramp up their chatter while others dial it down, keeping the group's overall activity the same. It's learning, basically, Rumbaugh says. That redistribution is defunct in mice that lack a copy of SYNGAP1, the new work shows. Only the decrease in activity happens, resulting in a weakened ensemble overall. It really shows that SYNGAP1 is playing a role in the mature brain, says Kimberly Huber, professor of neuroscience at the University of Texas Southwestern Medical Center in Dallas, who was not involved in the work. The neurons in those ensembles could be divided into three groups. The team found more than half of the cells were weakly active during whisker stimulation, about 30% were moderately active, and about 7% were highly active. As expected, the overall activity of the ensembles did not change in the wild-type mice, but the distribution of the ensemble's activity did. The weakly active cells scaled up their firing, the highly active cells dramatically suppressed their responses, and the moderately active cells showed little. Similarly, in the SYNGAP1 mice, the highly active cells grew quieter, and the moderately active cells held steady. But the weakly active cells did not ramp up their signaling after the new experience, Rumbaugh and his colleagues found. That's the twist. And it has a complex effect on the circuit, says Richard Huganir, director of neuroscience at Johns Hopkins University in Baltimore, Maryland, who was not involved in the study. By contrast, neurons from mice that lack a copy of SYNGAP1 weaken their connection with cells that fire out of sync but do not strengthen their connection with those that activate at the same time, suggesting that the gene is essential for this mechanism. That suggests that the potential window for treatment of people with SYNGAP1 mutations extends beyond early development, Huganir says. It also points to the possibility of harnessing SYNGAP1's ability to strengthen synapses and improve learning in other situations, Rumbaugh says. He and his colleagues are looking for compounds that can boost SYNGAP1 expression. Please support my channel to grow by pressing subscribe button and the bell icon. We will notify you technological news. Thank you.